开讲吧。好，等一会儿啊。
你把你那个先关了声音，然后我开声音，思考在我这边，可以听到我说。OK，首先感谢大家来今天在百忙之中来参加我们的这个 mini lecture series 活动，然后也谢谢 Steven 和 Sean 准备了很长时间 for this presentation. Now I'll hand over to Steven.
对呀，然后大家好，我们这次就是我们的第一个 lecture series，就由我和 Sean 我们两个一起来进行这个讲座。然后呢，今天主要讲的是这个 stem cells in cancer。然后首先就是大概部分是我先介绍一下整个 stem cell，就是 cancer stem cell 的 research。然后 Sean 他会讲一个比较细的一个 case。然后先是看 background，然后你就直接翻。对，首先就是我们知道 cancer。我们有一种 cancer 叫做 metastatic cancer， 就是说这些 cancer 它们会在体内进行转移。那有一个很好的问题就是为什么有一些 metastatic cancer 它们转移之后，你进行相应的治疗能够治好，但有一些又不行。然后我们首先再看一些 evidence。Only a small portion of tumor cells from colonies in vitros。它的意思就是说，我们有的时候在我们取的一个 tumor 那个出来的话，只有一些很小一部分的这个 tumor cells。它们可以生成一个 colonies， 就它们可以自我复制。大部分的 tumor cells 它们是没法自我复制的。那下一个，那 large numbers of cells must typically be translated to form tumors in xenograft models。就是说，它需要很多的这个 cancer cells， 它们要转移到另外一个体内才能进行这个自我的繁殖。那大概是多少个数字呢？就之前有统计，在很早之前，他们有做过一个 research， 就是说一般来说至少要转移100万以上的这个 cells 到另外一个体内，才有可能在那边繁殖，就才产生 tumor。那当然这个实验现在已经被禁止了，因为这是就是不太道德的，这个一般是在小鼠上面进行。那我们现在看下一个，就是说 in acute 这个 AML 这个 disease 呢，它是已经被证明了，就是说只有很小一部分的这个白血病，他们的这个细胞能够进行比较持续的这个自我复制，但是大部分都不可以。所以说问题就是在于我们现在看到的问题就是说 how do metastatic cancers develop？ 首先第一个 all cancers can proliferate but only small numbers can actually complete process。就是说所有的这个 cancer cells 呢，它们都可以就是进行转移，但是呢只有很小一部分它们才能真正完成整个过程。Only a number of anatomically divergent cells can significantly proliferate to make two new tumors。只有很小的一部分它们才能大量的自我复制，然后呢产生一个新的这样的一个 tumor。
大家觉得是哪一个？是一还是二？
对。
猜对了。
然后我们继续看，然后我们现在讲讲这个 stem cells 和 cancer 之间的关系，就直接先放。对，你就把这个对。首先这个我们就在 intro bio 上面学过，这个 stem cells 呢，stem cells 呢它就是它叫干细胞。这干细胞呢它能够就是转成 progenitor cells， 然后呢并且就是这个 genes 它可以分成各种各样的这个细胞，每个细胞呢它有自己不同的工作。但这个 stem cell 本身呢，它的目的就是为了能够复制，然后成为不同的这个细胞。然后然后那个 chemotherapy 就是还有另外一点在讲的是关于 cancer 的，就是说现在传统的这个治疗 cancer 的一些方法，就比如说单纯的化疗呢，他们的治疗手段是说，去攻击那些自我复制速度特别快的细胞。但是这样它有一个问题在于什么呢？就我们比如说指甲，然后头发这些本身自我复制速度就比较快的这个细胞，它们就会受到攻击到，但它们本身是健康的。所以看到很多化疗的人做 chemo 的人，他们都要把头发剃掉，因为到时候后面头发会光掉落，很可怕。那首先这个 cancer 这个 CSC cancer stem cells 它是怎么做的呢？我们现在看一下这个 stem cell 的组成。首先 stem cell 它本身是可以分裂成一个 stem cell 加上一个 specialized cells， 也叫这个 progenitor cells。然后这个 progenitor cells 呢，然后能够等会它继续在自我复制的时候，它可以变成各种其他的 specialized cells， 比如说看这边这个neuron 也可以变成这个 astrocytes。然后 stem cell 一个很重要的一个能力就是 self renewal， 就是它能够自我产生这个更多的这个 stem cell， 这个是很重要的。然后我们现在看这个 cancer stem cell 它的 research 整个 timeline。首先这个 cancer stem cell 这个 idea 是一开始是在那个1997年 John Dick， 然后和他的同事一起在这个 U of Toronto 发现的，在1997年，他们在这个白血病里面发现这个 CSC cancer stem cell。然后呢，接着在2003年的时候呢，这个 Michael Clark 在 Stanford，他也展示了出来，就是大量的这个 cell，这大量的这个细胞呢，在这个乳腺癌里面，他们是不能够进行就是自我繁殖、自我继续生长的。然后他们同时也就发现了，就是在这个 breast tumors 里面也展现出了有这个 cancer stem cell，就说只有一小部分这个 stem cell 它们可以自我复制，然后呢，就是变成更多的这样的 cell 的这个现象，一个 evidence。没事没事。然后在04年的时候呢，这个 Peter Dirks 同样也是在 U Toronto，他就是发现了类似这样 stem cells 的这个细胞在这个 human brain tumors 里面。接着05年的时候呢，他们在这个癌细胞在骨癌就 bone cancer 里面呢也发现了这个 stem-like cells。然后呢，这个是 Parker Gibbs 在这个 University of Florida 发现的。然后呢，在现在做 research，到现在。然后我们看一下整个这个 cancer stem cell research 它整个这个市场 industry 里面的一个前景。就我们可以看到这个前景就是预计未来是每年会以12%的左右的这个速率在往上持续增长的。然后呢，这个 stem cell market 它其实就是说，在这个 cancer stem cell research 的这个投入，然后和它这个能够完成的这个therapy 的这样的一个比例，然后它的一个投资。然后我们可以看到，就是在全球的话，这 cancer stem cell 它主要的这个target 的就是个别的 cancer 呢，就是主要是 breast cancer 和这个 blood cancer，也是 leukemia。对，然后这个我们是看到这个有多过21个国家，他们都有在就是做类似相关的研究。好，接下来的话就是由这个 Sean 他会给我们讲一个比较细的一个 paper，讲说这 cancer stem cell 的一个 research，然后我们来看一看。对
OK，
Unmute yourself.
all right, so I'll be discussing this case study called Prospective Identification of Tumorigenic Breast Cancer Cells. This is a paper by Al-Hajj in 03, so it's pretty early. Essentially, we discuss breast cancer, right? Stephen mentioned how breast cancer was one of the bigger drivers of the cancer stem cell market. And that's because most of the research done on solid tumors for cancer stem cells have been on breast cancer. So we're just going to go straight to the approach, the background, like we already discussed. Breast cancer is one of the most deadly metastatic cancers out there in the human body. And we've had this sort of discussion / debate on whether tumor cells are able to all of them are able to replicate, or only a small phenotypically diverse bunch of cells replicate and then form new tumors. And it has been demonstrated in acute myeloid leukemia or AML, so blood cancer, that it has been the latter that works, but no work so far as of 03 has been done on solid tumors, and this is what this paper sets out to find out. So whether in a solid tumor that we have a small bunch of cells that would take up the function of forming new tumor colonies. So the approach, as illustrated over here, we take samples from primary human breast cancer cells, and then we implant them into mice that has been immunocompromised. So it's on the mouse mammary fat pad. So basically where the breast is, and we prep the mice with estrogen, etoposide, and anesthesia with ketamine. So etoposide specifically here is what does the DNA damage. It is a topoisomerase inhibitor. So our intro bio folks should know what topoisomerase does. Relieves the stress on DNA on a replication fork. So etoposide essentially destabilizes that and causes DNA damage. So mostly double-stranded breaks or DSBs. And so yeah, so after the tumors have been, the human tumors have been implanted in the mice, they are then dissected, and we take out the tumors, the new tumors that have formed in these mice, and we do a flow cytometry on them. And I'll talk about that shortly. So this is just like a general overview of what happened to the mice. That is, and it's not really related to the results. It just shows you like what has been done to each mice. There's tumor T1 to T9. These are the actual tumors that was taken out from humans. And then obviously it says origin. So it's either still like an isolated tumor in the breast, or for everything that's other than T2, it's metastasized. So they were able to, take out cells from the tumor from all over the body, right? And for our first passage or first formation of the tumor in the mice, all of them made new tumors, right? And then all of them were passaged in the mice, meaning that the tumor was taken out of the mice and then re-implanted in new mice. So I'm just going to briefly talk about flow cytometry and FACS. This is probably, if you're working with cells and you're working with anything in like cell bio or Moleq, you're going to run into flow cytometry and FACS at some point. So I think it's really useful for me to just kind of like give you an overview of how flow cytometry and FACS works. So we want to start with a cell suspension on the right there. Ideally, it's going to be a single cell suspension. So that means no clumps, no like cell death, and everything is stabilized. Everything is like in a straight line. And then we just have one cell at a time passing through the flow cytometry apparatus. What we want to do here is we want to stain the cells for specific markers. That could be markers on the surface, markers in the cell, fluorescent markers mainly. And then once we stain these cells with fluorescent markers, we pass the cells through a light sensor one by one. So one cell at a time, right? And then for the different like markers, if we shine lasers on them, and then if they return a signal of some sort, then we know the cell has been expressing one protein or some other molecule within the cell. And then here's where the difference between flow cytometry and FACS comes from. FACS has one extra step, right? It has the sorting, which is where the S-chem comes from. And so you use either electricity or some other sort of method to physically separate the cells once they've been identified based on what markers they have. So for example, like you can attach like a really negative ion, on your marker itself, so that when it marks something, and then you pass it through this electromagnetic field, it's gonna stick to one side, and then that perhaps leads to like a different tube than your other samples. So that's how flow cytometry and FACS works, and that's what the paper did. So here we're gonna look at these flow cytometry graphs. Essentially, what we're looking at here is CD44 and CD24. And what they were able to show is that these are, so by the way, like you see T1 up, that means unpassaged. And so that's the original tumor that they got out of the patient, and P means passaged, right? So, so I think the graph itself is pretty self-explanatory. Up that way, y-axis means more CD44. And then going right, we see more CD24. And then the dot plot essentially just shows like for each cell how much CD44 or 24 has been expressed. And then the boxes on these graphs are essentially what the researchers are interested in. And that is CD44 plus or positive and CD24 minus or negative.
Good question.
Yes.
What do CD24 and CD44 like? What
are
they tags for?
What are they? What?
Like, are they tags for something? Like,
oh, they're tags for the immune system. Right? T cells look at these and they go like, okay, like this is what we want to.
Is there a specific cell type associated with CD24 and CD44?
They're T cell markers. T cell markers. Yeah.
44和24不同是在哪？
是我们不知道它不同在哪里，就是说它的这个目的只是说我们看到这两个 marker 有这两种 marker 本身，我们不知道这个 marker 在做什么，但是它能作为一个 group 的一个标签。对，我也对这个问题我刷过很久，就说 like what do these markers actually do? The answer is we don't know, right? But we were able to characterize cells based on these.
Oh, okay,
yeah.
Right.
Yeah, and so for all these different injection densities of tumors, we start from the low end over there on 100, and then the highest is 5 × 10⁵ power. we're able to see that like this little group of cells called CD44 plus, CD24 negative, ESA plus, I think, is what really does the job, right? So like even for like the really low concentrations, you're able to see that like 10 out of 10, 4 out of 4, that means mice. Like that's the number of mice out of the mice injected that formed the tumor. So for that specific group, we're able to see that like whenever we find CD44 plus, CD24 negative, and then I think ESA positive is what we're looking for. Yeah. Those are the cells that consistently forms new tumors in new mice. Yeah, and then this is just like an additional thing that they not proved, but they showed evidence towards, and it's that DNA, so the cell cycle isn't really affected, right? So we don't see this group of cells like replicate like crazy or do anything within the cell cycle that is different from normal cells. So the cells over here at A are the actual tumorigenic cells. The graph B are normal cells. But you can see that like they have the same amount of cells in the G0, G1 phase and S, G2, and M phase. There isn't really like a huge difference within the number of cells that are in either of these cell cycle phases, which means that none of them are like, prolonging their DNA replication or like doing something weird within their like meiosis or mitosis cycles. They're just their cell cycles are completely and then over here we have some histology proof that histological proof that like CD24 negative cells when implanted in mice have a specific like obviously more pronounced tumor formation than their CD24 plus comparatives.
What is DNE?
DNE are stained samples for the for tumor cells.
So
and then more red means more DNA. Yeah, so it's like more tumorigenic. And then over here we just see that like so A, C, and E going that way are actually from two different mice, right? So what the researchers did was for these CD24 negative but high ESA cells over here, they used a box to isolate these cells. So they want these cells, right? They isolate these cells and then they throw these cells back into a new mouse. And then when they tested for flow cytometry in that new mouse, we see that there's a bunch of new cells that have like all sorts of different like CD24 and ESA expressions. What does that mean? That means that the cell has phenotypically proliferated and they've generated a selection of different phenotypes, which really speaks strongly to the hypothesis that it's a small group of cells that do all the work when they form new tumorigenic colonies, right? And we sort of see the same here, which is that like for CD44 high and CD24 low, which is what we screened for originally, when we isolate these cells and then put them in a new mouse, flow cytometry tests that new mouse, we see a bunch of different expressions for these cells as well. And so, yeah, what are the takeaways, right? We've had the ability to separate tumorigenic and non-tumorigenic populations of tumor cells, and that should allow the molecular characterization of these cells. And so, CD24 and 44 being one of them, but also maybe we can find other markers that also separate the tumorigenic cells from the non-tumorigenic ones. And that is able to help us to elucidate the pathways that account for the tumorigenic potential. Because that pathways we still don't know, right? Like if we actually knew the pathways and then we're able to develop something for those pathways, then we can probably just cure cancer. And then the other thing is the prospective identification of tumorigenic cells might have prognostic significance for patients with breast cancer. And so what that really means is we can now test for these markers, right? Like if we see somewhere else in the body other than the breast, we see, oh, like there's a lot of like CD44 positive, CD24 negative cells. What does that mean? That means you probably have metastatic cancer, right? And so, yeah, so that really just speaks to the prevention of relapse of tumors. So you get tested for these markers once in a while. If you see like an abnormal jump in one of those markers, then that's probably bad news for you.
question.
Yeah.
Do you need to do a biopsy to, like, 就是你要怎么样才能 test these markers like other
that's a good
place,
question,
like,
yeah.
然后每次我去医院，然后呢要把我的肺也割一块下来，然后呢我就看有没有
我觉得应该是抽血。对
学界的话就是说去看 marker，
对
然后来做。在 biopsy 的话，它可能会
，biopsy 一般都是有，就是能看到肿瘤了，CT 上面看到有肿瘤了，然后再去切肿瘤。
这样说把它割下来，然后你放在箱子上，然后根据它的 differentiation rate，还有就是它可能你会 stand 不停的 marker 上面，然后再根据那个结果来。
OK，
我就把血放在一个，
就像你培
就是
养
OK，
对对
Yeah.
So yeah, do we have more time to like go on? Sure. 但是
就是我把
哪个？
再往下一个。
像这个啊，
就是我刚刚听到这个术语，因为我不太了解这个方法。
啊，
就是你们的唯一的 validation that 这个东西能够 cause cancer 是 only 2 mice, right? You do only 2 mice to prove that this is actually
没有，就是这个只是他们 repeat 了很多遍，但是这只是 like 可能应该是 like 就效果最好。对，Yeah。行，那讲一个问题，要不讲？
那我问一个问题吧。
你问吧。
Going back, so this is all like， 就是你这篇 paper 是03年的嘛，对吧？那在这过去的20年之中，有 any like new findings on this topic?
I'll have to find out. Yeah, I'll have to find out about new findings. Yeah.
就是所以，因为我们现在是知道 CD24和44都是非常重要的肿瘤的 marker， 是这篇文章或者就是这个时期的文章带出来的一个结论
是的。
哦，OK。
就说他们只是看到了现象，但是说就他看不到本质，如果真看到本质的话，我们的治疗癌症就不可能。
这是有问题。
就是我不知道后面是不是还有就是我想问一下 therapeutics cancer stem cell therapeutics 就目前有哪一些？是现在还在 basic research 阶段，还是已经有一些
Lisa 能不能讲一下
我其实因为我虽然做 stem cell，但是我不是很了解 cancer stem
？Cancer stem，
但是我可以讲一个我知道的，就是因为我之前做的是就是有一个 translation initiation factor 嘛，然后现在有一些就是pan translation initiation factor repressor， 然后就是用来治疗癌症的，就是因为它可以去控制它的这个protein translation 的 rate， 就是因为这些 initiation factors 它在 stem cell 以及 cancer stem cell 里面它比较表达的比较多一点，所以我们可以去抑制它们来就是控制这个 cancer stem cell 的一些就是 proliferation rate。
这跟biopsy 都挺像，就是我们当时很多 case study 都很像。我的比如我们 lecture 能够就跟你说把我 lecture 能够讲比较好玩就好，不太激烈。
我要不要 start 一个 new Zoom meet because it's ending?
Sure.
Okay.
Stop share.